All right, so as Pradeep said, it's over to me now. But everybody kind of stretch for a minute so you don't start to get the Zoom fatigue early. The goal of this session is stretch, do yoga. The goal of this session is to introduce you all to just some of the labs that are kind of participating in this event, as well as, you know, get us to know each other, because we're largely a local community, and so we have asked a few of the PIs to introduce their work, and so we're just going to try to do about a 10 or so minute short burst presentation of what each lab is studying by the PI, and then have some periods for questions. So first up, we have Frank Yi, who will give us a taste of the amazing diffusion imaging work he's been doing. Frank, it's all you.
Yes. Hello, everyone. So let me share my screen here. So my lab is mostly doing diffusion MRI and tractography. So for those of you heard of this techniques, it's using MRI to detect microscopic diffusion movement. And using that movement, we could tell what the overall connection looks like and create some kind of pretty picture, like just like what my background is showing. We call it tractography. So to give you a flavor of it, instead of just talking about it, so once we get the diffusion data, this interface, this is a tool developed in my lab. So what diffusion MRI is doing is, first of all, we get the data and we calculate some kind of diffusion distribution. So just that I showed here, across the brain structure, we know like for each voxel is like a microscopic road of diffusion pattern. And by analyzing or modeling this pattern, we get a rich set of informations like fiber orientation, like myelination, like a lot of different things. And from analyzing those information, say for example, showing the lab, we could see, okay, for each voxel, there could be fiber going this way or that way. And you could, from those fiber orientation, we could plot the road, we call it a fiber track. So that's why we call it fiber tracking. So to give you a flavor, they're doing in real time, that this is a whole brain fiber tracking, just using those information we got it from diffusion MRI. But of course, the challenge here is how could we be so sure that this result is reliable? How could we validate it? So a lot of huge efforts from the community using machine learning, statistics, MRI techniques, all kind of way we try to get the most accurate result of it. That's the major focus of my lab, and this has been the topic for the past years. And also talk about my most recent research. So for those tractography, it has been around for like decades, but right now I'm moving forward to like new tractography modality. So in the past, when we get those pretty tractography, then we move on to analysis. But it's always like, okay, we get a track first and then do the analysis later. But I thought it's like, is there a way we could combine both together? So instead of finding difference in track, we track the difference. So it's kind of integrating analysis and fiber tracking all together and try to show the results. So the picture showing in here on the upper left is like just a comparison. The inside figure A showing the conventional tractography, we show the whole brain pictures, but those tractography is not sensitive to subtle difference. Either there's an injury, either there's a demyelination, tractography still showing them. But the inside figure B is showing that, okay, we only track those with difference using longitudinal data. So it's kind of like masking out other regions or other tracks that has no difference and just show those change over time. And I call this like differential tractography because it's showing only the difference. So instead of finding all the track together and then analyze it later, we can target the result, target the difference directly. And follow the same paradigm, we can also do something called correlational tractography. Say, okay, we have a good study, you have like a study variable, you want to know which pathway is correlated with something, a behavior score, or either just a group difference, or any kind of scale. Then we could track correlations instead of, okay, finding track first and then study correlation later. We track the correlation directly. So to give you a flavor of it, I also have a data that we used in the past. This is the data from team's lab, 60 subjects. And what we're doing is we correlate at the voxel level, correlate the diffusion pattern with BMI. So we could get a T statistics that says, okay, if the BMI goes up, the diffuse and the sorter feeder integrity goes down, then we could plot kind of a map in the lab. So you can see the intensity, higher intensity means that it's more dropping as BMI goes up. And then what we could do is that we're adding this information and then feed into the tracking pipeline. So here I turn on, okay, I want to track the decrease of it. So if you see that it giving the fiber tracking algorithm like a additional mask. So instead of tracking the whole brain, now we could focus on those 3 pathways with a decrease when the BMI goes up. So we call it the correlational tracking. So right now, once I keep the fiber tracking, it will show only the regions that decrease as BMI goes up. And this we could combine with multiple regression, postural correlation, any kind of multiple regression model, and then try to just see or study the pathway or study the structural function correlation. And we found it to be a very powerful tool to study the circuit mechanism. Go back to the page. So the picture showing here is our recent paper. We applied this to a group of stroke patients, and we try to see what's the circuit mechanism that involved with aphasia. And this shows which pathway could be positive correlated or negatively correlated. So that's one of the major focus, try to innovate new tractography modality instead of just mapping the pathway and do an analysis later. Then the second focus in my lab is try to building up a template and also an atlas. So we want to try to know what's the standard floor plan of human brain. So what we have been doing is we either mapping young adults or either different age group from Human Connectome project. We try to see what are the major pathways. So it's a heavy collaboration with neurosurgeons, neuroanatomists. So what we end up with is that we could average thousands of subjects together. So we, first of all, we create a template. This from the Human Connectome project data. So this template is kind of averaging all the diffusion pattern across thousands of subjects. So for each muscle is already in the MRI space. And we would know, okay, for most of the subject, what's the fiber orientation goes in that direction or another direction. And then we could do fiber tracking. And once we do a fiber tracking, we invite all the neuroanatomists, neurosurgeons coming in, helping me to say, okay, this is a language pathway, this is visual pathway, this is motor. And so to create and reach information of atlas providing to the community. So it could be used in any kind of analysis or even just understanding the human brain anatomy. And this will be the second focus in my lab. And the third focus is trying to understand the structural function correlation. Here we propose something called local connectome fingerprint. So it's like a very specific quantification approach to using diffusion MRI to see what's the connectivity at the voxel level. And what we found is that it's very highly specific to each individual. So it has high specificity to the unique connective pattern in each subject. And what we're hoping to do is that we try to correlate this with any kind of function, behavioral score, and try to understand the circuit mechanism behind this. And of course, my lab is focusing on tool development to help neurosurgeon before they go in. So right now we're routinely doing this for brain tumor surgery. So as we map those pathways for tumor patients, we'll provide this rich information to neurosurgeons. So before neurosurgeon going in, they know whether the pathway has been pushed up or down or inside or been destroyed. So they have better information to avoid important pathway. Say for example, like language pathway, motor pathway, they can avoid it. So that's an overall view of what my lab is doing. Any question here?
Thanks, Frank. Anybody have any questions for Frank on his diffusion imaging work?
I think
I'm
Frank
biased,
is attending.
by the way. I'm biased. I think it's great, but I have a conflict of interest there.
Yeah. So Frank, you're attending the whole hackathon, right? Today and tomorrow.
Yes.
Yeah. So
like a project using the local Connectome fingerprint. So the idea is using those diffusion information to see if we can predict subject's answer to personality questionnaire. So say, for example, if the subject answer and I'm a worrisome person, they will get a 5 number of scores and see if we can use microscopic diffusion pattern. That's the structural type of information to see if we can predict fixed behavior.
yeah. So he'll be around if you guys have more questions or interest. You can always chat him up during various socials. Yeah.
Okay. So I'll do a little switch in the schedule here, just for the time some people have to be here. So next up is going to be Baya Luna, who does amazing work on the cognitive neuroscience of development and its role in diseases. So Baya, it's your show. Take it away.
All right. I'll share my screen. Is that being, no, never mind. There we go. Is that good?
Yep.
Yes. All right. Thank you so much for doing this in Pittsburgh. I think Pittsburgh is really rich for being able to apply these emerging approaches to data analysis. And my lab is particularly interested because of the work that we're doing. And I'm just going to tell you very briefly the type of work that we do in our lab. I know a lot of you are also familiar. And also to incentivize those who might want to, participate in some of our studies in the hackathon this afternoon and tomorrow. So our overarching goal is to understand what is underlying the transition from adolescence to adulthood. We are particularly interested in this because of two main aspects. One is it is, or you might remember from when you were a kid, that adolescence is a time of increased sensation seeking that can undermine survival and actually increases mortality to a great level. So that, like, what's going on in the brain that might be underlying this. More importantly, though, I'm in a psychiatry department, and most major psychiatric disorders emerge during adolescence. So something critical is occurring there that we need to understand. That's why things like Andre's talk is incredibly important for us because we have so many variables that we need to bring together to try to construct this model. We also collaborate with Frank, who is just amazing, and with Tim and with other people here. So we can really leverage a lot of this information. So we obtain a lot of information, as you will see. So here again, is just to tell you that the adolescent period here is completely unique and distinct from earlier development and later development. And this is what we're trying to characterize using multimodal neuroimaging. So of course, we always include behavior. That is what we're trying to explain. Why are there differences in cognition? Because in adolescence, you've reached a level where you have access to adult level cognition, but you're not used in a reliable fashion. And this is something that we need to explain. Of course, we use all the typical, task, rest, structural fMRI, but we've also used MEG in the past. We are now using a lot of spectroscopy at 7 Tesla. I'll tell you a little bit about that. We use PET and we use tissue iron. And of course, we are looking at white matter structure using diffusion spectral imaging. So one of the primary things that we're trying to understand is what is the state of the executive cognitive networks in comparison to the limbic motivational networks, because this is, for the most part, what is characterizing the adolescent period. And we have a model called the driven dual systems model, where what we propose, as opposed to previous models that you might be more familiar with, because the media likes to think that prefrontal systems here presented in the solid line are not available in adolescence, but aspects of motivation are predominating. Our model, based on the data that we have gathered for 20 years and those of others, really indicates that adolescence is a time when prefrontal systems have come online, but they're driven by motivational reward systems in an adaptive manner so that you can gain the experiences needed to specialize into adulthood. So let me tell you, of course, we've been doing this for like 20 years. I'll tell you a little bit about what we are currently pursuing to understand this. And first, we are very interested in dopamine. As you know, dopamine is underlying this whole motivational and reward aspect of behavior. And when you look at the animal literature, and we really like to stay close to animal models so we can understand mechanisms in our neuroimaging data. And we know when Bart did a review of the literature that there are many different aspects of dopamine, and each one of them seems to be having a different developmental trajectory. But adolescence is, again, something that's very unique in what might be occurring. Typically, what we see behaviorally, like I said before, is an increase in motivated behaviors. And we want to understand what is the role of dopamine in this transition. So we have this one, study that we're about to finish. We have like 2 people left. We're just waiting for the scanner to open again. Using the MMR machine, where we can get both PET and MRI at the same time. Using PET in pediatric populations is difficult. It's not impossible, but it is difficult to do. So what we did was the following. We had a cohort of 12 to 30-year-old subjects. For these subjects, we did a bunch of MRI acquisitions, including R2', which is specific to being able to measure tissue iron. We could have used QSM, but that's a lot longer, and R2' seemed to work very nicely. Why did we care about tissue iron? Some of you may know, some of you may not know, that when you think of brain iron, you think of blood, but there's tissue iron called ferritin. And this iron is predominantly found in the basal ganglia. And it has been found, it is a very important step in the development of dopamine. And it's been looked at for a very long time in Parkinson's literature, restless leg syndrome, ADHD, to get a feel for striatal neurophysiology, but no one's really been thinking, what is it saying specifically about dopamine? So in any case, we obtained that in all our subjects. And then from 18 to 30, we additionally, and at the same time, we're also obtaining PET measures. And we obtained measures of presynaptic dopamine availability using DTBZ and of postsynaptic D2, D3 receptor density using raclopride, because like I said before, we don't know which aspects of the dopaminergic system is changing with development. So here are some of the results, and this is important for a lot of the work that we're doing now going forward. So first of all, as other people have found, we find that tissue iron is increasing with age. In the ventral striatal limbic area, we find that it's actually starting to even stabilize when it's going through adolescence. Importantly, we found that this R2 prime tissue iron measure was very nicely associated with the presynaptic measure of which is a more direct measure of dopamine availability. So this is big because it's telling us, okay, you can actually now characterize in a non-invasive manner a very important aspect of dopamine function. So this has become extremely important and very revealing. We have this in Nature Communications. It came out a few months ago and so forth. And a lot of people are now really becoming interested in this tissue iron. In addition, there's this R2 prime acquisition of tissue iron. It's great in many ways. It does have limitations because it's sort of new. And all of these fancy processes and pre-processing aspects that have been developed for fMRI are not available for R2 prime. So there's a lot of, to look at every subject, do, quality assessments. You get rid of a lot of data because it's very sensitive. So in some aspects, it's not as practical as, for example, leveraging the T2 star that you get every time that you acquire an API. Be it task or rest, you have that T2 star. And if you time average all, throughout the whole time course, that T2 star, you get a really nice measure of tissue iron. And what I'm showing you here is our latest. I mean, this literally came to me yesterday, but we've been looking at it for a while, that there's a very nice association between this more direct measure of, this quantitative direct measure of tissue iron and the T2 star that you get from, these things that you already have in your data. So now you can go back and mine all your old data. You can look at big data sets and start extracting this sort of information. So that's what we've been doing. Ashley Parr is going to be part of the hackathon today. Is a postdoc in the lab, and she already has a paper now under review and progress in neurobiology. Actually, she's responding to really nice reviews. And here she was really interested in looking what is the role of dopamine in frontal striatal connectivity and its changes in development. And she's also working to see how this is related to inhibitory control. So just very briefly, what I'm showing you here is some of the results that she has in her paper. It's available in bioRxiv as well. And something that other people had found at different ages, we really provide really conclusive evidence that the connection between ventral striatum and ventromedial prefrontal cortex, so the circuitry that supports reward learning and decision making, is decreasing with age, and that is great. It really goes with our model of specialization, that there's actually less connectivity. We interpret this that maybe that supports an attenuation of the influence of motivational systems on cognitive systems. Then she also found that greater amounts of tissue iron were actually related to weaker frontal striatal connectivity, supporting what we're trying to find developmentally. And then with the mediation analyses, she was able to found that tissue iron was actually mediating these decreases in frontal striatal functional connectivity. Now, the next step is let's go into big data and see what's going on there. So, of course, many of you may already know about the Adolescent Brain Cognitive Developmental Study. It's a multi-site. I think there's like 24 sites. We have a site here. I'm one of the co-principal investigators of the Pittsburgh site. There are 11,000 participants recruited at 9 to 10 years of age, and then they will be followed until they're 19 years of age. So we're talking about tremendously huge data. And even within that, there's a subset that that's called the ABCD Social Development Study, where there's about 5,000 of these individuals that get even more assessments that tell us about things that are related to delinquency and to those aspects of behavior so that we can start being more translational. And this is something that Ashley is very interested in doing, and she's already been collaborating with the National Justice Institute and so forth. So opportunities that we can do this. And she'll be looking at this tissue iron. She's already started to look at it. There's already some really interesting findings, predominantly, sex differences at early ages. So some of the other things we want to look at here are, we're going to be looking at how tissue iron is associated with pubertal changes, with DSI structural connectivity, with magnetization transfer ratio indices of cortical myelination. We also have a project where we're using past data to look at tissue iron in newborn imaging, and then how that's predicting cognition at 5 years of age, and how stress might be mediating that relationship. And then more recently, we've started to talk, because one amazing finding that I don't have enough time to talk about is that developmentally, one impactful characterization is that variability is decreasing, and variability that people think of as noise may actually be exploration, so that you can actually refine in the proper manner. So we're going to be looking at tissue iron and how different aspects of variability with FALF, but also with EEG. And finally, and I hope I have enough time, another aspect that's very rich in our laboratory is understanding the brain mechanisms underlying critical perioplasticity in the adolescent brain. This was spearheaded by Bart Larson, who's now a postdoc at UPenn with Ted Satterwhite's group, where we kept talking, people say, oh, adolescent, it's a sensitive period. And we're like, wait a minute, maybe it's a critical period. And he went and into the literature, wrote a beautiful paper, really identifying that all the molecular mechanisms of critical perioplasticity are evident in adolescence in prefrontal cortex. And just very briefly, for those who do not know about critical perioplasticity, the visual cortex is a prime example. You're born, your eyes open, tons of light comes in, that makes all this excitation go up relative to inhibitory systems. Excitation, reflected by glutamatergic function, inhibitory by GABA. And then the whole, the architecture reorganizes so it can bring these two back to homeostasis, a homeostatic balance. So what we, primarily postmortem studies and animal models have shown that, in fact, in the human brain, in prefrontal cortex, and also in animal, there are these predominant changes in GABA and glutamate. So we said, okay, can we go and look at this? And I was talking to Hobie, this is what I would like to do. And he's like, actually, we can do that, but you have to go to 7T. And that's primarily because GABA, which you might see here, is this little blue thing. You can hardly see it. So at 3T, it's very difficult to discern this signature of GABA, and it's very contaminated by the macromolecules. But with 7T, of course, you are increasing that signal to noise to a level where you can actually see this. And you can actually start to separate and really identify what's GABA and what's glutamate. So that's what we're doing. We're using MRSI, and this is led by Maria Perica, a graduate student in the lab. So unlike the typical voxel spectroscopy, this is a whole slice that provides us with a bunch of voxels throughout that slice, and we can get the whole spectrogram to understand what's going on with GABA and GLU. And our initial findings are the following. We had really hypothesized that GABA would go up and glutamate would go down in prefrontal cortex. But instead, what we're finding is that GABA is stable and glutamate is taking a nosedive. And, discussing this with our colleagues doing the postmortem work, they are not surprised that this is what is occurring. The question is, though, does this have any of an impact in this balance between GABA and GLU? So we've been applying an approach that's already in the literature where you're actually correlating at the group level GABA and GLU. And the more that they're correlated, the more this is telling us that these two functions are starting to become balanced. And in fact, this is what we're starting to find. And these are correlations, thus no SEM bar. I always freak out when I see plots without SEM bars, but this is the case here. And you can see that the balance is increasing and showing dramatic changes when you're going from adolescence to adulthood. So this is mind-blowing that this is what could actually be happening. And we're actually finding that there is evidence of this critical perioplasticity in adolescence is starting to close in adulthood. By the way, the closing is not complete, and there are closing mechanisms like myelination. That's why MTR and DSR are so important. And perineuronal nets, which I wish we could image, and I'm sure that one day we'll be able to do that. So what we're doing now with this is we have a bunch of correlates. I'm really enthusiastic about combat, and I want to see that talk again by Andre and actually reach out to him and so forth. We have so many correlates that we want to look at to try to make sense of this critical perioplasticity. So we have a big range of cognitive assessments that we do with eye movements and with computer games. We have psychological assessments to see, even though this is a normative sample, and I didn't mention in this cohort, we're looking at 10 to 30-year-olds. We have this MTR view of myelination obtained at 17. We also look at EEG because we're interested in these changes in variability that I was talking about, but we're also interested in getting an understanding of how signal to noise ratio at the level of EEG, which is a more direct measure of neural activity, is coinciding with this regain and balance of GABA and glutamate. So, we have a fantastic lab. Everybody is taking chunks of this. We have people that want to visit from other labs and do postdocs to really mine this data. We always make our data freely available through the NIH. So people can go and play around, and I hope people are enthusiastic to help with us. Finn is the co-director of the lab. He is, the analytical mind, knows everything, and he'll be giving a pitch for one of these studies that we're interested in participating in the hackathon, and we'll be providing oversight on how this happens. So thank you very much. Thank you for inviting me to participate.
Thank you, Baya. As usual, just a tsunami of work. That's phenomenal. Does anybody have any questions for Baya? I know there was a lot in there. I actually had one for you, which is a lot of your work kind of tries to balance a gap between the low-level microscopic neurobiology and molecular biology with the macroscopic imaging that you can get at this big scale. What are some of the particular challenges that your lab has had trying to make this mapping from the micro scale to the macro scale? Because that seems to be relative, that to the topic of the brain hack today, trying to figure out ways of doing this mapping and removing noise. What are some of the biggest challenges your lab has had on that?
Well, I think one of the biggest challenges that people who do developmental work are aware of is that there are, certain sorts of artifacts in neuroimaging that are very age sensitive. For example, head motion. And a lot of these and other sort of artifacts that we constantly have to be looking into, even with GABA and glue. And, you know, particular challenges not only with age, but with hormonal differences. So we also obtain hormonal, you know, through hair sampling. So we have real hormonal type of stuff, and we start to see differences like that. On the other end, Tim, it's really about careful interpretation of what we can and we cannot see. Of course, with 7T, we are getting a nice measure of GABA. Is it really the GABA that's going into another synapse? Not entirely, right? There's free floating GABA. So there's a real, a lot of work that goes into, after everything's done, what can we actually say? And for me, it's very important to not, you know, when we write papers, we don't just throw it and say, okay, you interpret it. You know, bite the bullet. We say, look, this is what we think is going on. Based on this, we might be wrong, but this is what we're trying to understand. And that's how we start to build this model that we hope is important in being translational for mental illness. For example, doing a growth chart. So I was super excited when I saw this growth charts coming up. We've been talking about this for a long time, so that we can really start to understand, you know, a pediatric neurocognitive growth chart, so we can start to predict those kids who are at risk and be able to help them.
Vince. Oh, actually, I'm sorry, Vince, we can't hear you. I'm sorry, Vince.
Oh, and Vince does really great work. I think really highly of what you've done, Vince, and what you're doing. And we have a collaboration with Vince now in the newborn sort of projects, and I'm sure Ashok will touch upon as well. And Vince, we can talk anytime.
Oh no, yeah, the sound driver crashes are always the Zoom pain. I'm sorry, Vince. Maybe we can come back to it in a little bit once the sound card works. Baya, thank you so much. We all appreciate it. I think we should move on for the sake of time. Unless Pradeep, did you want to say something?
No, I was gonna say, you know, for people like me who don't have much neurobiology experience, what you touched upon today sounds like a great topic for a bit of a review tutorial, especially the Bot Larson review you mentioned. You know, there's an unconference session tomorrow. If there is anybody who has the time and expertise, they can teach us, you know, what we learned from all this work. Which is
Okay, yeah. What time is that, Pradeep?
2 p.m. I believe. I can double check the.
Yeah, I'll check the schedule. Thank you, everybody.
Thank you, Baya. All right, next up is Ashok, who is the driving force of neuroimaging at Children's Hospital and radiology here at UPMC. Ashok, the floor is yours.
Okay, you can see my slide?
Yeah,
okay.
Yeah, we can see it.
So I'll just quickly go over what my division and my research group at Children's have been focused on. And I am keeping it very broad, not getting too technical. I've a couple of members in the lab who are here participating, but I was going to quickly go over some of the more broad topics that our group has been covering. So let me just, okay. So our Pediatric Imaging Research Center, we started it about 10 years ago. And our goal was to provide advanced imaging support for different clinical pediatric translational needs. Some of these needs are truly on the clinical side, while others are more related to research. So we have, we really are a combined clinical and research operation, including now having two magnets, a Siemens Skyra and also a Siemens Prisma, that are both used for clinical and research. And these are just some of the breadth of the applications, which are not restricted to the brain. Our group is, I would say, is neurocentric, but we do provide support for even other kinds of imaging research needs outside the brain too. And so in terms of our own research focused on the brain, we do have a lifespan approach where we have imaging that we do from the fetal period actually all the way to adult. I actually have to update the slide to reflect that. So we recruit patients to study normal development, but also look at populations, and I'll talk a little bit about that. So a lot of our technical needs arise because there is not one approach to acquisition and post-processing that fits across the lifespan, unfortunately. I wish it could be that case. Fetal imaging itself, as you see here, has its own challenges because of the fetal motion and the limit of tissue contrast that we can extrapolate from the fetal brain. And then as you go through the first month of life, there's rapid development of multiple brain metrics, including myelination, which also raise lots of technical challenges for us from a quantitative aspect, but also on the acquisition side. And then things start to stabilize out once we get into late infancy, childhood. Obviously, there's lots of continuing development that happens right through adulthood, but some of those technical challenges tend to go away. So I always show this as the sympathy slide, I guess, because we are very interested in critical brain injury. My personal research interests that I've been studying since 1995 has been white matter injury that is seen in newborns, particularly in preterm babies, but also in babies with critical congenital heart disease, which has been the shift of my particular focus over the last couple of years. And we do recruit these patients at different time points. We do assess them with different types of neurodevelopment. Obviously, the assessment of a infant, where we do Bailey's and other kinds of testing, is very different than our older kids where we're using toolbox. And our batteries are very broad because most of the children, preterm children in congenital heart disease patients that are surviving now have a very broad, they have deficits in multiple domains. Specifically frontal executive, social behavior, memory. So our batteries tend to be pretty broad. And then we combine that with neuroimaging to address questions. So in terms of my particular focus in the group, even though we function as a very broad core to provide support to the research community of children's, I've been very focused on the congenital heart disease cohort, which we know these patients are now, because of the improved cardiac care, actually survive now all the way to 90 years of age. And a lot of our questions have actually shifted over the last 20 years since I've been studying the white matter injury problem, where our focus was initially on surgical outcomes or surgical techniques that are used in the perioperative period very early on as potentially driving outcomes, to a much more genetic environmental type of question. And there's also now a concern for early dementia in this population. We do use different types of innovative techniques that other people here have probably spent a lot more time developing to be able to answer some of the questions that we are interested in, which is mostly predictive models of outcome, but also understanding modifiable risk factors. How do they affect the brain? Because that helps us develop interventions. So this is one particular grant that is leveraging Connectome technique to understand outcome in a prospective longitudinal study of 500 children with hypoplastic left heart syndrome, which is a very rare disorder. Some of the technical challenges for doing this type of work is because of these rare disorders and these, we typically have to scan now multi-center, which brings a whole level of harmonization techniques that our lab has started to move into so that we can harmonize imaging data across sites. We have also moved into developing clinical trials now where we're trying to take some of these imaging biomarkers and understand, as neuroprotective techniques have come out, especially in this particular study, we're looking at the developing white matter vulnerability. And this is a particular clinical trial that is currently on hold right now because of COVID, where we're using dexametastomidine, which is a low-cost sedative agent that can protect from the effects of hypoxia and also anesthetic toxicity in the developing white matter. And in this particular trial, the imaging is actually used as the outcome measure, and then patients are followed further for behavior. So I do think we're in an era where a lot of these innovative imaging techniques are going to be used as not only surrogate biomarkers of prognostication of behavior, but also treatment effect. And that's really an area that we have been moving into. And so a lot of the harmonization techniques are really critical for us to be able to do these clinical trials in rare populations like congenital heart disease, where we would have to recruit across 15 sites. This is just moving now more to the end of the lifespan, where we do have concern now of our cohorts, both preterms and congenital heart disease patients being at risk for dementia as these populations are being followed out. And so we have started collaborations looking at genetic exposures in the adolescent period where we think there's vulnerability, where what we initially considered dysmaturation and abnormal development may actually start becoming early accelerated aging. And I think the adolescent period in the congenital heart disease cohort may be that critical period where we see these changes. Our genetics collaborator, Cecilia Lowe, who's the chair of developmental biology, we have been starting to get into different unique ways that we can start matching different kinds of phenotype. This here is a nasal scrape that we do that allows us to look at the motion of cilia that is in the nasal cavity, which we think is prevalent in all organs of the brain, including the brain, and all organs of the body, including the brain, kidney, and other structures. So this is a particular way that we can map looking at different types of phenotype to the genotype. And these are some of the additional studies that we're undertaking. And we have moved into looking at now how the ciliary motions actually relate back to different types of dysmaturations mapped with cortical thickness and other kinds of imaging techniques. And just another example of how we actually also combine imaging and genetic type of outcomes and vulnerability going to metabolism. We've been very interested, and this is the slide that Chet Mathis gave me, which actually might be from one of Annie's papers, but of the vulnerability of Alzheimer's in the posterior default mode. And this is an area that we are also concerned about in the congenital heart disease population. So we have been undertaking with Dr. Lowe's lab some mitochondrial studies, or iPSC, where you can actually pull the blood of the patient and study the metabolism of mitochondria in the blood, and then compare it to metabolism in the brain using spectroscopy and phosphocreatine, other techniques. We also do sequence development. So this is a project that Raf Cheston and Vincent Horst and our group have been working on. We are putting a new server in inside the firewall at Children's with the Prisma to allow us to now move into more rapid ways of imaging where we combine not only the innovative sequence acquisition, taking advantage of dead space in the sequences, but combining it with novel imagery construction at the scanner. And this is very important for our studies, as we would like to be able to scan as fast as we can with the best resolution so that we can avoid sedation and anesthesia in the pediatric population. And then lastly, I am involved in developing different types of clinical translational imaging across our lifespan in our department in Pittsburgh. And so there's a lot of interesting work that's being done out there in multiple departments. And I just wanted to, this is a new center that we just started related to artificial intelligence that Shandong Wu is leading in our department. And there's a lot of opportunities for collaborations across multiple schools, particularly for people interested in applying machine learning to medical imaging. And I'll just stop there. I know I kind of went through a lot really fast and kept it pretty broad.
Thank you, Ashok. That, as always, you've got your hands in a bunch of different projects, but I really like the breadth and scope of your work. Does anybody have questions for Ashok? I have one. So, you know, I find it fascinating that you can try to build these pipelines that look at development starting in utero. And, you know, even if you skip the in utero step, what are some of this unique analytical challenges that you have trying to track brain development with neuroimaging from even, you know, postnatal days through adolescence?
I think the biggest challenge when you start applying a longitudinal question, especially when you're going from fetal to neonatal, a lot of our approach is in acquisition, but also the different types of segmentations and the way that we process DTI, the way that we look at functional connectivity is very different than in the prenatal period versus the postnatal period. So there is a whole host of questions. And this is a very critical, this transition from fetal to neonatal or prenatal to postnatal is probably one of the most critical areas of brain development related to genetic vulnerability. And so I think a lot of our challenges are right around that area because we're switching from very different acquisition and different types of tools. The motion is also very different between the fetus, obviously the placenta, which moves in a different way compared to the postnatal brain. And then in the first year of life where we have quite a bit of rapid brain development, there are similar challenges because your tissue contrast between T1 and T2 is changing very rapidly. But I do think we have some new initiatives, including Baya and myself are involved with the new HBCD study where we're trying to tackle some of these challenges to acquire data. But there are quite a few technical challenges.
Seems daunting, to be honest. Yeah.
It's extremely daunting. Yeah. And the more we learn, the more challenges that come up. So.
Well, thank you, Ashok. I really appreciate the presentation. As always, if you guys have questions for any of our kind of labs or any of the speakers, feel free to reach out during some of the social times too, throughout the event as well. So just wanted to make it clear that don't have to consider your kind of questions now, but if you'd like to discuss, we have the Gathertown meeting area, which we'll move to next. But moving on, thank you, Ashok. I'd like to introduce Annie Cohen, who is a researcher at the Alzheimer's Disease Research Center and studies basically the neurobiological markers of Alzheimer's disease using multimodal imaging. It does just absolutely phenomenal work trying to apply what I think are creative neuroimaging approaches to trying to understand Alzheimer's disease. So Annie, the floor is yours.
Thanks. Just to be sure, you guys see the full screen slide and not presenter view, right?
Correct.
The two screens always mess me up. Okay. So I'm really glad to be here to talk to you guys a little bit about the work that we're doing. I thought I would focus rather than trying to summarize the past 10 years or so of what we've been working on in terms of PET imaging and development PET tracers, I thought I would focus on some of the challenges that we currently have with the imaging work that we're doing, because I think there's a lot of creative minds here that might be able to tackle some of these issues. So just as an introduction, when we're talking about the pathological cascade of Alzheimer's disease, we're talking about a cascade that starts at least a decade prior to the emergence of clinical dementia. So these are when people show up in the clinic complaining of memory problems. The changes in the brain, particularly changes in the proteins amyloid beta and tau, are happening years before we see those clinical symptoms emerge. And so what our research team's interest has really been for many years is, how do we detect that pathology using neuroimaging at its earliest stage? So when you're talking about changes in amyloid beta, which is, according to the amyloid cascade hypothesis, believed to be the first change in Alzheimer's disease pathology, how early can we detect that in the brain prior to the emergence of any clinical symptoms, which are out here in this sort of green line? And then there are these other changes that can be detected by neuroimaging that come somewhere in the middle. So we're talking about subtle changes in cognitive performance, subtle changes in glucose metabolism in the brain, which you can measure by FDG PET, subtle changes in blood flow in the brain that you can measure using fMRI or ASL, changes in tau pathology, which is a relatively new PET imaging technology. So it's only been around, it's been around for less than a decade. And so these are sort of all the things that we're interested in looking at in order to, one, better detect Alzheimer's disease pathology earlier, and two, determine what sorts of moderating variables there might be that could modify that pathology or modify your risk of developing dementia in the face of that pathology. So our research group as a whole looks at a variety of factors: social determinants of health, cardiovascular disease, cognition in midlife. So there's a whole series of research questions that we ask that I won't get into, but that's kind of the goal of our research studies. So these neuroimaging techniques have really been developed in terms of Alzheimer's disease to have better diagnosis of the disease. So you have to remember that before PET imaging technology was developed to see amyloid pathology, you needed pathological confirmation at autopsy of amyloid plaques and neurofibrillary tangles. So the development of this technology has led to better diagnosis of Alzheimer's disease, and then has allowed for preclinical diagnosis of AD and clinical trials in individuals who are pre-symptomatic but have Alzheimer's pathology. And for us, on the other end, or on the end of that spectrum, on that preclinical spectrum, we're asking questions about what sort of features in midlife into old age might impact those pathologies. So these are just to kind of orient you to what we're looking at and what we're talking about when we're looking at both amyloid and tau imaging. So this is a series of amyloid scans from individuals who, all elderly individuals, some who are cognitively normal, some who are cognitively normal with amyloid pathology in their brain, some who have mild memory problems, which we refer to as mild cognitive impairment, or MCI, who have different levels of MCI or amyloid pathology in their brain, from essentially no amyloid pathology all the way up to very significant amyloid pathology, and then Alzheimer's disease. But a couple of things to note here that I think are important in terms of the technical challenges that we face is that even in our negative cases, so in this first panel and then in this MCI negative case, you see white matter binding of amyloid tracers non-specifically. So these are binding, we think, likely to myelin basic protein rather than amyloid. And so you see this sort of white matter trace with the tracer. What's interesting is one of the ways clinicians are taught to visually read amyloid images is that distinction disappears. So you no longer see that trace of white matter because there's so much cortical uptake of the tracer. But this white matter binding can be a challenge, this sort of non-specific binding that we talk about. So what about tau imaging? Well, we still don't know a ton about the longitudinal aspects of tau imaging that we understand for amyloid imaging. We know that across amyloid positive Alzheimer's disease cases, there's a whole gamut of uptake of this tracer. We know from postmortem studies, so correlations between tau imaging in life compared to postmortem tau staining, that there is good correlation between the tracer and how much tau is in the brain. But one of the things that we're learning is that there's a lot of variability in the amount of tau uptake. There are also challenges that I'll talk about in a minute with off-target binding of tau tracers, that they bind to structures in the brain that we know don't contain a whole lot of tau, and we're still trying to tease apart why that is. One of the challenges with tau imaging that we don't have with amyloid imaging is that amyloid plaques don't exist in the white matter. So it's very easy to say that that's not specific binding. But some of the areas where there is non-specific binding of our tau tracers are also areas very close to areas where there's a lot of tau pathology. And so this gets a bit challenging. So that leads me to sort of our first challenge, and that's off-target binding. And so this first image up here comes from a really great paper that was just published in neurology, I believe, outlining some visual techniques for rating scans as tau positive. So one of the important things that all PET imaging research really needs in terms of Alzheimer's disease work is to understand what's pathologically significant uptake of a tracer and what may be meaningless. And so this is, while amyloid imaging, this has been an established field for a while, with tau imaging, there's still a lot of work to be done. And what I wanted to show here was some of these regions of frequent off-target binding. So you can see in the scalp and bone, there's very high levels of off-target binding. In the striatum, particularly as individuals get older, there's more off-target binding. There's a debate in the field. The hypothesis is that this is likely binding to calcifications in the striatum, and so the older you are, that may be an issue. The biggest problem with these tau tracers is that they bind to the choroid plexus. They frequently bind to the choroid plexus. The issue with this is that there's a lot of tau in the hippocampus. The hippocampus sits inside the ventricle where there's choroid plexus lining all around it. So it's been a real challenge, and very frequently we just throw out hippocampal data because we can't distinguish whether we're looking at binding of choroid plexus or binding of hippocampus. And so one of the real needs in the field, and I think one of the things that that needs to be done, is to figure out how to, at a very high level, segment out hippocampus from choroid plexus, and then be able to apply that to our PET images. And that's something that sort of eluded most people in the field and has moved people away from this particular tau tracer, which is now FDA approved, to tau tracers that don't bind as much to the choroid plexus. But that doesn't change the fact that people have data from thousands of individuals using this tracer. So we really need better methodologies to distinguish choroid plexus from brain structure or from brain gray matter, so that we can determine what sort of, where there's real uptake of the tracer. The second challenge that I wanted to talk about just very briefly was this idea that, particularly when you're dealing with brain aging, pathology doesn't occur in a vacuum. it's going to be, you're often going to see comorbidities, co-occurring pathologies. And this can be problematic from both the standpoint of understanding what's driving cognitive problems. So are you talking about cerebrovascular pathology in the brain pushing cognition, or are you talking about Alzheimer's pathology? But it also creates technical issues. And so this is from a paper that a medical student who worked with me, Anna Goodhart, published several years ago, showing that when you have significant white matter hyperintensities in the brain, so shown in the bottom panel, you can see that there's significant sort of loss of PIB binding in those areas. So the tracer actually doesn't bind to these white matter regions where there's lots of white matter hyperintensities. The challenge with that is because PET is so low resolution, you actually may be losing information about cortical uptake as those white matter hyperintensities creep into, at least from an imaging perspective, cortical gray matter. And so this can create challenges with understanding if someone's amyloid positive or amyloid negative, and sort of regional analysis of amyloid. The second thing I wanted to mention, and this has been, or the third thing I should say, this has been a big issue for our group, is the fact that cerebrovascular pathology in particular impacts free surfer segmentation. So white matter hyperintensities and strokes in particular can impact segmentation from free surfer. Historically, in our amyloid imaging group, we hand drew regions of interest. As we started scanning more and more individuals, this became not practical for high throughput. And so we moved to, as most of the field did, automated free surfer processes. And what we found was in our individuals, like in the previous slide, who have high cerebrovascular pathology, that free surfer had a very hard time segmenting those brains. It became very, very difficult to, for us to get proper region of interest placement. We've subsequently found that high levels of brain atrophy also affect this free surfer segmentation, and motion, which I'm going to talk about in a minute. And in particular, in our Down syndrome studies, where we have a very hard time getting individuals to remain still for both the PET and MRI scans, motion has become a really big challenge and something that's really slowed us down and caused us to lose a significant amount of data. So we found that we can hand edit most of these cases, except this really, really, truly severe cases, but it's taking our technologists upwards of 12 hours, sometimes 24 to 48 hours to hand edit one severe case. And so we really need the development of sort of better methodologies for motion correction, either while in the scanner or post-scan. And we also need the development of sort of better segmentation methods that can handle some of these pathologies that are so common with aging. And so the last thing that I wanted to show was just an example of these really bad motion scans from our Down syndrome, one of our Down syndrome studies. So we rate every single one of our MRIs that come through in our Down syndrome study because this is such a common problem. And it's a 3 to 0 scale. So 0 is really bad, requires lots of hand editing, may not be rescuable, versus a really good scan for the Down syndrome study. And I don't know if you guys can see how, you can obviously see how bad the motion is on the, okay, there we go. How bad the motion is on the scan rated as a 0, but also how bad that free surfer segmentation is. It makes the analyses almost impossible to do. And especially in our Down syndrome studies, where we're talking about a very precious resource, very hard to recruit individuals, losing these data is losing a lot of time and a really precious resource. And so I think we need to do a better job, and with our colleagues who are developing methods, to think about how these methods work when everything isn't perfect. And I think that's a really critical piece of this, and kind of what I wanted to highlight, at least here to this group, because I think this is a group that can probably tackle a lot of these issues. And so I'm sure I took more time than I was supposed to, and I'll just stop here and think, obviously, this is just a subset of our collaborators. Our team is awesome. And we always say one of the reasons that we've been so successful is because we really like working with each other and with the participants. So thanks for the time.
Thank you, Amy. That was great. I think you bring up a very important point that, is obviously the focus of this brain hack, but it's something we often ignore, is that there's this assumption of the inherent validity of the recording that you get. And, that oftentimes people who particularly do pathological analyses looking at different patient groups ignores the potential confounds that are pretty obvious there. In your case where you're talking about the hand corrections, where you have, this intensive human effort to kind of do those corrections, do you have a large enough labeled set where you have pre-post corrected work that you might, we might be able to train methods to look at? And if so, how big is that data set?
Yeah, so we probably, no one's ever asked that question before, but we probably do, because we have two cohorts that have been historically problematic for us. One of them being the Down syndrome cohort because of the motion. The other being our oldest old cohort, not because of motion, but because of cerebrovascular disease and atrophy. Mm-hmm. And we probably have 200 to 300 images that are pre and post from those. So I don't know if that would be a big enough data set to train, but it's a really good question. That's exactly why I thought I'd present our data in this way, because I think that those are the ideas we need. And Pradeep says free server is almost always wrong.
Yeah.
Yeah.
Go ahead.
Yeah.
No, I was just, sorry, go ahead, Pradeep.
Yeah, even in healthy subjects, when everything is great, I think people don't understand how complicated a task that free surfer is trying to do. T1 weighted MRI is rather easy to deal with compared to other modalities, but fully automated reconstruction of the whole brain, talking about gray white matter, and gray matter CSF contrast, it's really, really hard. If you talk to image processing researchers to do that, it's a fascinating project, but it is almost always wrong in my opinion, because I've QC'd hundreds of subjects in the subcortical and cortical areas. I can tell you there is always a couple of OTCs that are where the contour is wrong. So we assume that we QC'd well, but it's never the case.
I feel like free surfer is like democracy. Like it's the worst form of automatic segmentation, except for all the rest.
Yeah, excellent actually analogy. Yeah. And what Anne tried to show is why it gets even more challenging in pathology. It's an important thing people haven't been looking at.
Yeah, and I think so. My gripe like with the Human Connectome project, which we've been running for almost 5 years. This is one of my gripes is if I need to get 16 minutes of bold from a participant who's demented, it's just not. That's not from a human perspective. When we're trying to bring real world people from out in the real world and not just healthy astronauts into study, those are not realistic things to do. So I think we need to get better. And this is, I come from the recruitment sort of neurobiology side and not the machine learning methodological side. And so it's easy for me to say, well, we just need to get better at how to process these things. But I do think it's a real need if especially the Alzheimer's field, is serious about studying real people and not just our ADRC retired professor populations who would lay in a scanner for 6 hours if I asked them to. Though, you know, that's just what they do.
Thank you very much, Annie. Actually, that gives us a good segue. So going from the applied, you know, real-world patient population to the machine learning side, that gives us the transition to our next speaker. Seong Jae is a professor in computer science, correct, at the University of Pittsburgh, who studies the applications of machine learning tools and computer vision tools to medical imaging. So, Seong Jae, it's all yours.
Okay, let me see if I can. So the right view, I'm guessing.
We see the
Okay.
split. We see the presenter view.
Okay. Now it looks good.
Perfect.
Okay, great. Yeah, I'm really happy to share some of my recent works. I'm Seong Jae. I'm a very recently joined assistant professor in computer science department at Pitt. So it's been about a year. So I'm pretty junior, and there's a few things that I've been working on since I got here. And so I'll mostly be talking about just a couple of projects. But before, so to share a brief introduction about what I've been doing, computer vision, machine learning, deep learning methods is my specialty. I got PhD in computer science, and I've been using structural MRIs and structural connectivities, and sometimes amyloid and tau PET scans. And I've been working closely with Dana and Annie and Daphne from School of Medicine at the University of Pittsburgh, and wanted to give special thanks to my students who've been basically responsible for and made all the works that I'm going to talk about possible within the last year. So want to kind of, hopefully it's not like an in-depth discussion, or I don't really have much of an experience to cover a lot of like broad things like everyone else has been doing and will be doing. So I want to hopefully from my talk, I wish people can kind of think about if they can adapt or maybe apply some of the things that I will be talking about to their approaches or to their pipelines or problems in case they're facing similar problems. So the underlying premise of the works is that what happens if you have multiple sites and multiple scan data sets. And sure, people already know that harmonizing data is, I would say it's more of a data perspective of dealing with the multi-site, multi-scanner problems. And I'm going to talk very briefly about it. It's joint work with Dana, and we have a very special data set of 1.5T and 3T scans from 16 people that are at most 3 months apart. And the second one that I'm going to talk a little bit more about is how can you actually enable machine learning, complicated machine learning models and deep learning models so that it can actually be applied to scans from data sets or sites or scanners that you have not seen before. So this is more of generalizing a model. So one is to deal with this problem from more of a data perspective. The other is more from the model development perspective. So the first topic is I'm going to spend like a minute or two on it. We have a very unique data set of where each of the 16 normal aging subjects have 1.5T and 3T T1 weighted scans that are at most 3 months apart. And we're going to use free surfers to derive cortical thicknesses and volumes. And hopefully they should be similar, assuming that the biological variabilities between these two scans are minimal and free surfer is doing its job. But there are a lot of things going on that we realized that we found out that these two measures, which should be pretty similar, are oftentimes not similar. So there are two approaches that we're considering right now, and it's still an ongoing work that we're putting up a manuscript with Dana. First is to use a well-known combat harmonization technique so that, and it's very possible because you end up getting a set of measures. So you start out with images, we can end up getting a set of measures, and they're going to harmonize them. So this is a more standard way, I would say. And then what Dana has been thinking is that, well, since these are based on images, what happens if we perform something on the images and then derive these measures? So one of the things that we've been doing is apply some sort of a normalization technique on the images, and then derive these measures and see what happens. So this Ravel normalization that came out 3-4 years ago normalizes the intensities of the images. It's similar to some of the other normalization techniques on your imaging data sets, such as white stripe. And then we are going to use the same free surfer and then derive these measures. So it turns out that I'm not going to show the results or anything here due to the time, but this also happens to help out with the harmonization situation. So we're kind of trying to solve a harmonization problem with a normalization. And we're currently analyzing it, trying to get neuroradiologist ratings and so on. So how similar it is to different data set scanners. I'll get back to it. So we're currently working on it. And as you can see, we can actually combine these two pipelines, right? You can have these images go through normalization and then get these measures and then go through the combat harmonization. So we're kind of holistically looking at everything and analyzing and so on. So hopefully this goes out there and then we'll have a preprint and so on. But just wanted to kind of end at this point because I want to talk about this other thing. But if you're more interested in this work, there's a very similar work that our collaborator has done on Down syndrome population. So same method has been used, but with a slightly different analytic pipeline. Want to take a look, you can check this out. Oh, thanks, Annie. So just want to move on to the more machine learning, deep learning aspect. Similar idea, except we're not using the same data set, but still neuroimaging data set. Now they're coming from multiple sites and multiple scanners. So let me, I want to kind of describe the problem first. So typically in machine learning or any sort of a training and testing pipeline, you have this one big data set, and you're going to kind of divide them up into training and then use the remaining as a testing and so on in a more of a cross-validation manner and so on. So you assume that there's one big data set, and I'm going to just do my thing within that data set. Realistically speaking, what if you have a data set that consists of scans or subjects from multiple sites or multiple scanners? Then within the one data set, you have different distributions of data sets. So that's kind of the problem that I want to tackle. It's a situation that I think is going to be likely to happen more and more in the future, where, so I found this very interesting data set online. It's a white matter hyperintensity challenge data set, which has 20 scans from 3 different sites. So Amsterdam, Singapore, and Utrecht. And this is our local data set, not part of this data set, but so the question by looking at this data set, I thought is that, well, what happens if you train a model on a subset of these scanners and then test it on the other one, except the one that you test on, you never look at him during the testing. So for example, you're going to train a model on Amsterdam and Singapore, and then never look at this Utrecht. And then once you train on these two, you're going to test it on it. What's the guarantee? How do you know that your model is going to quote unquote generalize well? And I think this could be a very realistic scenario where you train a model on a single site or a data set from a single site. And if you want to deploy it in the real world, you need to be able to prepare for a very stratified cross-validation is very important. What happens if you just don't have the samples from this new data sets ever? That's kind of exactly, and that's kind of a very extreme case that I want to see if this wanted to see if I can tackle. So the experimental setup is quite kind of synthetic in a way that I manually kind of came up with a combination to make this scenario happen. What was, oh, interesting. I'll take a look at that, Pradeep. And there is a very analogous problem in computer vision and machine learning. It's called a domain generalization, and where basically the same idea, except the terms are slightly different. So you train on these data sets or samples from specific sources. So in this example, you can imagine that you have images of dogs and elephants that are in photos or art paintings or sketches. So you're going to only train on these images. And then your model right now is able to classify it between elephants and dogs. But based on photos, art paintings, sketches, what happens if you see cartoons of dogs and elephants? So people have been kind of artificially creating these types of setups to kind of try to tackle this domain generalization problem. Same thing could happen basically in a case of our data set, our multi-site, multi-scanner data set, where we can imagine we're using Amsterdam and Singapore images as sources, train some sort of a model. In our case, they're going to do a wide matter hyperintensity segmentation, and then train on this data set. Again, you never see anything from this data set, but then when you deploy it, hopefully it does some reasonable job. So what is it that I have been trying to do? Specifically, I've been trying to develop a white matter hyperintensity segmentation method. So automatic segmentation of these white matter hyperintensity regions. And this could be a potential model prediction. And the problem setup that I created is that we have four domains or 4 data sets, and going to just come up with several combinations to make this happen. So we're going to train on Amsterdam, Singapore, test on Utrecht, train on U and S, test on A, and so on. And then finally, we're going to train on AS and U, and then test it on the PIT data set. So we all have, all these data sets have manual tracing, so like the ground truth labels, except the PIT does not have like a full slices. So that's why we kind of have a separate setup for this one. So just to give you a very brief idea of the results, when I say no DG, it means like a simple unit, normal unit that does a reasonable job when you just train and test within the same data set. That should do pretty well, and it's been doing pretty well. So no methods, no extra method has been done to perform this type of a setup. Now, normally they're okay, but there is a specific case where you train on Amsterdam, Singapore, and test on Utrecht, it just fails miserably. And it's not like we intentionally try to make it fail. We perform some reasonable normalization that other people would do in like typical setups. So same setup, same training setup, except different data sets, they usually work well. But we immediately found a case where it just does not work well. And just by looking at it, you could tell that this data set, though from Utrecht, they had like a more variability. I think they had like a really weird, like distinct histograms if you compare it to others. But when we applied the method, that I will be able to talk in details, it's not that complicated. Same model, just trained differently, is able to, we've realized that it's able to actually turn into a more robust setup. So same setup, same model, just trained differently is able to improve it pretty significantly, I would guess. We're not looking for like a huge improvement, I just wanted to avoid this worst case scenario, basically. And the last setup is that when you train on these 3 data sets, Amsterdam, Singapore, and Utrecht, and test it on PIT, we also found some improvements. Still preliminary, but I think this kind of gets the message across. And this is also a part of the white matter hyperintensity segmentation challenge. It's a public challenge, and I think it's working pretty well, as we're currently ranked 5th on it. And that's mostly because no one really cares about this challenge anymore. But so the main point is that in practice, I think this is a very likely scenario. It not only happens in medical imaging, it's a very well-known problem in computer vision, and people are trying very, very hard to make sure and prove that the model is robust in very extreme cases when you have samples from distributions or data sets they have not seen before. And the second thing is that it's not just a matter of adding more stuff to the model. Depending on how you train it, I think, is able to add some robustness to it. So that's kind of what I've been working on for the past 1 year since I got here. Hopefully it was interesting. If you have any other questions, you can ask me now or send me an email to me, Dana, or Davneet, or check out my website to see what I have. Thank you.
Fascinating talk, Sanjay. Fascinating. I think we
Thank
have
you.
a lot of common interests here, So yeah, it's going to be probably a lot of discussions. I won't take up all the time, but one thing I quickly want to mention is the point of stratified cross-validation. So like you mentioned, ensuring that the distributions in training and testing are similar, or understanding how the differences in distributions among the testing and training actually changes the way you evaluate or how they perform is actually very important. And although machine learning community tries to focus it on a theoretical perspective, when in reality, as in practically speaking, each domain has very different properties that may not really, that what we see in, for example, conferences like NeurIPS may not directly translate. We need to actually do actual data mining in real neuroimaging data sets to understand how it happens. So I would say we need to focus on our own domain as much as we try to learn from the computer vision people and NeurIPS researchers, right? So yeah, one thing I've been wanting to do for some time regarding this stratified cross-validation is basically enabling people to test this in an easy manner, you know, like you're talking about harmonization at the imaging level, which is actually requires a lot more challenging, a lot more what you call development. But even after the features are extracted, it just splitting them in interesting ways is not easy in nested cross-validation actually. So if anybody is interested, I want to pursue that either this hackathon or even later, you know. So anyway, probably we should let other people ask you questions on this important topic.
Thank you, and for your comments in the chat as well.
Tom seems to have a question. Come on, go ahead, Tom.
Not really a question so much as just it's a general problem of how to make algorithms that apply to unseen situations that we recognize as humans, but the machine hasn't seen it before. Something I'm working on is automatic segmentation of whole slide images of histologic sections taken from Alzheimer's disease brains. And something as simple as just gray-white distinction on immunostains versus H&E versus silver stains is really challenging. And if you train an algorithm only on one data set, it doesn't apply to images scanned from a different case necessarily, or from a different institution. So it's exactly the same problem.
And just wanted to make a very quick comment. So yeah, I think everyone has probably run into this problem in one way or another at some point. And this has been pretty prevalent in the just machine learning community. So I've been talking about this very extreme case when you just have not seen even the samples. Now, what's a little bit simpler cases where you have the samples but don't have the labels, and that's a domain adaptation. What's even simpler, I would say, is like a case where you just have like one or two samples with the labels from this new domain, and that could be like in the realm of like transfer learning. So there's like a series of problems with different difficulties. What I just kind of described is like very extreme case, and people have been trying to kind of explicitly try to, not just in the neuroimaging, but mostly in computer vision and machine learning, try to just tackle this very, very extreme case. So I think that kind of we don't have to like use domain generalization all the time. This is just when you just, in the worst case scenario, this is what you would do. And that's why this problem is actually extremely difficult, not just, you know, application wise, but theoretically speaking. Like, you just can't make a lot of assumptions when you don't have anything from this new data set. So I haven't really mentioned the computer vision aspect of it, but I've been working on my student with a more of a theoretical aspect, like trying to describe like, in which cases that you can actually consider these methods that's out there with some guarantees and bounds and so on. And another quick thing is that that this idea is basically kind of similar to deconfounding models that people have been also coming up with. So if you consider, in this case, I was considering sites and scanners as like a domain that you want to kind of factor out. But then there are other problems where people have been using similar models to actually learn the presentations that are pretty deconfounded already by using similar models, so that when you put them together in this feature space, they're not like naturally distinct by their domains or whatever factors. They're kind of merged together and so on. So I think all the problems that we have all thought about it, I just kind of gave a very specific case. In case people haven't really seen it in detail. So I'm not really trying to bring anything new to the table.
This is fascinating. Actually, this actually reminds me of a fundamental problem across, I think, both maybe all domains of statistics, in my opinion. Something about defining the distance for your problem. You know, if you can find the right distance metric, you know, a lot of these machine learning problems and computer vision problems actually can be solved. You know, so in the middle of my PhD, I spent a lot of time trying to, if only I can tune the distance metric to my problem, maybe I can improve the classification. It's actually hard. I'll touch upon it in my talk, why I actually like multiple kernel learning in that sense, because I feel like, if you tune the kernel as in distance metric to the problem, a lot of other things can be actually solved down the line. We can't just blindly apply Mahalanobis distance or, just Euclidean distance to all the problems, right? So, yeah.
Well, thank you, Seongjae. It sounds like there's a lot of overlapping interests among the community right now on what you're doing. So I anticipate you're going to get a lot of questions in the follow-ups as well. Okay, in the interest of time, because we're coming up on lunch, and I know people get antsy around lunchtime. I do too. I'm going to go ahead and jump in and see here. Can everybody see my screen okay? Yeah, okay. And I have lost my mouse, but that is okay.
Tim, you said you would introduce yourself.
Yeah, so my name's Tim Versteinen. I'm a low-level researcher at Carnegie Mellon University. I don't do much. My lab does everything. So everything I'm going to be talking about is really no credit to me whatsoever. It's to the students and postdocs who did all the work. But you're listening to me present it. So I run the Cognitive Axon Lab over at Carnegie Mellon University, and our primary interest is on the structural constraints of cognitive algorithms. So what do I mean by that? Well, we all know that the brain is a giant network of interconnected neurons and glia that all have directed communication between each other. And this network isn't arbitrary. There's a particular structure and form to this broad network that gives rise to our behavior. So my lab is really fundamentally interested in understanding the architecture of these pathways, the specific unique configuration of connections in the brain, and how those constrain the algorithms of cognition. And what I mean by the algorithms of cognition are the high-level algorithmic descriptions of behavior that give rise to specific types of output. Primarily, we're interested in learning and decision making, and so that's where most of my focus is. So my lab tries to kind of span research that goes from network mapping to algorithms of behavior to even novel fundamental theories of behavior. I've been accused of being a Marian, and I think that's probably a reasonable summary of how I approach science. So first, I kind of want to give you a brief taste of some of the work we've been doing on looking at neural circuitry and its relationship to behavior. We've done a lot of work with Frank, and he presented some of that work as well as building anatomical templates of macroscopic connectivity. I'm going to shift a little bit and focus on something a little bit different. And it's still something that Frank touched on a little bit in his talk, which is our work looking at what we call the local connectome, which is the map of all the fiber-wise connections in the human brain. So using DSI Studio and methods that Frank presented earlier, we can essentially come up with a gigantic map of all of the fibers of every white matter voxel in the brain. And so what we do is we build an atlas that is a representation of the normative fiber patterns in the white matter of the human brain. And we can then use those as sampling points on orientation distribution functions, normalized version, which we call spin distribution functions, in every individual subject. And we can sample those in every individual subject. So what we do is we basically use the template to say, in this voxel, there's two fibers that are oriented in this way. We can take the spin distribution functions from every subject, and we can create essentially a fingerprint. So this is basically measuring the magnitude of diffusion density in every single fiber of the human brain. This is the 433,000 feature dimension feature, and it kind of encapsulates the microscopic local integrity of all the white matter in the human brain. And as Frank kind of pointed out, these fingerprints are unique. So here you're looking at 3 different subjects scanned at 2 different times, and you can see that the geometry of the kind of hills and valleys, the rises and falls of the fingerprint are reliable within subject, but reliably different across subjects. In fact, we can calculate the inter-fingerprint distance of each subject. So what we're showing here is the distance between fingerprints between every pair of scans. Some scans are the same subject, some scans are different. And the blue distances along the diagonal are the same subjects scanned multiple times. So you can see the clustering of the same subjects. In this case, these are the same subjects scanned 3 times. And we can use these distances as a simple metric for classification. So this is the distribution of within and between subject distances. And you see we get a cleared separation of these distributions. And using a very, very simple classifier, we're able to get reliable pairwise accuracy. So across 17,000 within subject pairing scans, or tests, we can get 100% accuracy on individual subject identification. This gives us a classification error akin to an actual fingerprint. So we have about the same error in identifying the same subject as you get with an actual fingerprint. So these are reliable metrics of the overall kind of white matter integrity of an individual subject's brain. So we moved on to doing work on trying to characterize the differences in local connectome integrity. And as I mentioned, this is a high dimensional object. So we might have, in our human connectome project work, we have thousands of subjects, but for every subject, we have about 433,000 features that we have to analyze. So this is a high dimensional statistical problem. So we adopt methods like sparsity constrained principal components regression to kind of compress the dimensionality of the problem. So we use principal components analysis along with a lasso constrained regression model on those components to build models of individual differences in terms of things like cognition, education, demographic information, anything you want your Y to be. And we can reliably build these predictive models of individual differences along an output feature dimension using the white matter local connectome as the input feature. And we've published a lot of work using these kinds of approaches. I'm going to highlight one that we just recently put up on the archive and is currently under review, led by Javier Rosario, who you'll meet this afternoon, where we look at how well the local connectome predicts individual differences in global cognition and compare it against other metrics like morphometry metrics from T1 weighted imaging, resting state connectivity, and stacked versions across all modalities to ask which features are most reliable at predicting individual differences in cognition. And we can generate these beautiful kind of feature maps like you see here, where we find that the local connectome, along with cortical surface area, are 2 of the most reliable predictors of individual differences in global cognition across individuals, but many different modality features actually contribute. So we have now this more holistic way of understanding how variability in white matter and variability in these morphometry and connectivity measures work together to kind of contribute and explain variance across individuals. So that's a very kind of brief summary of kind of what we're doing in the local connectome front. Because I'm trying to speed through so we can get through this, I'm going to shift gears a little bit to look at or tell you a little bit about our focus on the cognition front and the mapping between neural circuitry and cognition. So shifting gears here, we've done a lot of work over the last 5 years in collaboration with folks at the University of Pittsburgh Mathematics Department, looking at how we can map from circuit level architectures to behavior. So we don't have godlike access to the circuits of the brain that we're interested in, which are these cortical, subcortical, cortical basic ganglia loops, but we can build them in silico. So what we do is something called an upward mapping approach, where we take in silico spiking models of cortical basic ganglia loops that are as biologically constrained as possible. So this is a wiring diagram of our spiking network model. We can even add in plasticity like dopaminergic plasticity. And we have the model generate behavior as if it was a human or animal doing something like a bandit task, right? So a 2-choice bandit task. And we can model that behavior using cognitive models like the drift diffusion model. And what we can do is we can perturb the network in different ways and ask, how do perturbations in the network change the parameters of the cognitive model. And so this gives us a way of mapping between the microscopic dynamics of cortical and subcortical circuits to measurable behavior that we can then compare with humans and animals. So this is kind of a core part of a recently funded R01 in collaboration with Eric Guidry's lab as well, where we're going to be using these models to predict both behavior and physiology in humans and mice as well, where we have optogenetic control. And the nice thing about this approach is this gives us a theoretically principled way of predicting not just behavior, but macroscopic properties in the bold signal as well. So this is a study that I did with Kyle Donovan when he was a graduate student in the lab, where we use this kind of upward mapping approach to generate out specific predictions of what we would see at the output of the basal ganglia, the thalamus, if, let's say, uncertainty was modulating very specific properties of the decision process. So for example, if you become more or less uncertain about whether or not you should make a response, then the output of the thalamus should look like this inverted U-shaped curve here. And if on the other hand, uncertainty is changing something like the boundary height as opposed to the drift rate, you would get a dramatically different pattern. So we can use the computational model to make very specific predictions and counterfactual predictions to test different models of the underlying computational process. And indeed, what we find is that the dynamics of the thalamus shown here in these bar plots matches quite closely the dynamics of a model where uncertainty changes the drift rate. And we can actually map this kind of inverted U-shaped function across cortical networks known to be related to inhibitory control. So we're able to use these network models and kind of upward mapping models to build very specific predictions of dynamics that we should see in the bold signal. And very quickly, I just want to go over, we're extending this even further to just really understand the general nature of adaptive decision behavior. So this is that far end of that Marian loop I showed you, looking at the computational goals themselves. So we just recently published or put back up a preprint of a modified fundamental theory of exploration. This is led by Eric Peterson in the lab, where we have come up with a kind of a fundamental algorithmic way of thinking about information value and its role in exploration. So why do animals explore? How can we get this to be implemented in neural networks? And I'm not going to go through the details, but basically what we found is that we've come up with a fundamental theory that works around and resolves the exploration exploitation dilemma. It's a fundamental theory of how animals and humans and artificial agents can make the decision difference between exploring, finding information, and exploiting, receiving resources. And in putting this into an artificial neural network with this property, we actually have a neural network that can optimally explore. It can actually explore its environment in a way that has zero regret, maximizes rewards, and it does it better than any current state-of-the-art deep net that you can you get access to right now. So that's the goals of the COAX lab, this kind of structure function relationship. And I just want to acknowledge, like I said before, this is all the work of my wonderful team. Yeah, that is it. Any questions?
I have a question. That was great, really interesting. I was wondering if you guys have learned anything about the best way to stack different data modalities, because in my experience, sometimes counterintuitively you'll get worse performance, maybe due to the higher dimensionality.
Yeah, we've been doing two types. So Hobby's been using a nested variance of lasso. So for the stacking process, he's just using a lasso to do the feature selection and integration step. And that has worked fairly beautifully, because where you have redundant sources of information, the sparsity constraint is just going to pick one feature. So if I have two features that are equally predictive, there's no competition of their variance. We've also worked with using random forests as well. Those work particularly well when you have low sample sizes, because lasso won't converge in the tuning if you don't have enough observations, whereas random forest will work regardless. And that's where we actually start to see situations where your stacking will produce worse performance than any individual modality. And then, of course, it plays off with signal to noise. So where we have more robust predictions, we're less likely to see that stacking hurts us. But where we have very weak signal to noise, or really not that robust of a predictive capability, the stacking doesn't really add much. And in the preprint that I showed, that Javi just posted, we do actually have some output, some measures of cognition where stacking doesn't actually do better than an individual modality. So I think it depends on the nature of your question, as well as things like number of observations and signal to noise.
Okay. And when you say lasso, are you doing the PC? Are you talking about the PCR lasso, or do you do the PCA separately?
So for each channel, we generate up a single prediction using the lasso PCR. But then for the stacking of the predictions, we just use lasso.
Thanks.
Mm-hmm.
Fascinating, Tim. I'm really afraid to go after you. I didn't realize you're so big into simulations, man. I think there is a lot we can do with such a can we call it a generative model?
Yeah, absolutely. That's how we refer to it, as the generative models in the lab.
Right. Yeah. So there's fantastic approach. Yeah. I have a lot of questions, but I'll probably ask you later. Anyone else?
Around. Yeah, I have a question, Tim.
Sure.
So you have a spiking model that's making predictions about bold signal. How are you making that jump?
So we're actually using the intermediary model. So we have a spiking model that gives us intuitions about parameter changes in the cognitive model. And we can use the cognitive model, that accumulation of bound model, to make predictions about the bold. So all of our macroscopic predictions, whether they're bold or behavior, are actually principled off of the accumulation to bound models. So that's the way we do it. We're trying to figure out ways of linking more directly the spiking network models with bold. And I think that's going to have to work off of connectivity dynamics, because we don't have a full good understanding of neurovascular coupling across cortex and subcortical areas. And there's lots of work coming out about what's driving the bold signal in the striatum. Appears to be more dopamine than glutamate. So how you come from multi-unit activity to the bold signal is still a little unclear. But we can kind of proxy it by looking at connectivity changes, which would kind of ignore the nature of the neurovascular coupling in terms of evoked responses per se. And that's, I think, where we're going to head next.
I was hoping you were going to say there were advances and we understand that better now.
No, I thought we did. And then a bunch of papers came out of the Diceroth lab and then threw everything for a loop. So yeah, no. All right. Well, I'll be around if anybody has any more questions. And I know we are coming up on noon here. So Pradeep, I'm passing the baton on to you.
Thank you. Thank you. So let me share my screen. Where is it? Yes. Do you see the right screen now, Tim?
Yep.
Okay, great. So thanks everyone for joining. So my talk will be actually very different from what you've seen so far, where methods and challenges were focused on, like in terms of image processing or specific theoretical issues, etc. So I try to basically I'll try to present a very different fundamental problem that's actually outside the images, so to say. So before I jump into that, I'm gonna quickly show you where I come from, what my long-term goals are. So the biggest North Star, so to say, for me is to develop better decision support systems by something of a more general form for biomarkers based on quality, reproducible, and open science. By that, I mean basically in my opinion, most of our work is actually geared at offering better care at lower cost in a way. Most of this understanding into basic neuroscience, as well as better image processing methods or better predictive models, is aimed at trying to somehow improve quality of life and offering better care for patients. And this is where this is what inspires what I pursue, and why I try. It's important to bridge that gap, computational gap between the clinic and computing science people like us. So when I say decision support system, what I mean is simply any combination of patient data, in not just neuroimaging, but also electronic health records, wearables, and gait, speech, and whatnot, in combination with computers and domain knowledge from clinicians. That is either knowledge or data driven, or a form of both, to help make good decisions or better decisions, so to say. Often we think about it in terms of diagnosis, but it could be for treatment or monitoring too, but I focus a lot on diagnosis. Essentially in this space, what I realized over the years, as I'll delve into that in a second, there are two fundamental issues, so I realized those issues while trying to pursue what I call a domain-informed biomarker candidate search. For example, I think Anne kind of spent some time elucidating the challenges in terms of imaging. And what I try to do is to take basically the stereotypical changes caused by Alzheimer's disease and try to basically develop a feature extraction technique that catches those stereotypical changes. So we studied a number of different ways to extract various useful features from morphometric data as an anatomical T1-weighted MRI data in terms of atrophy, volumes, displacements, cortical thickness, or network level changes based on cortical thickness. And we develop methods basically that can be applied both to T1-weighted MRI, but also to FDG PET data. And to basically, the idea is to develop a technique that can actually separate disease from healthy at its core level, but it's a lot more complicated than, as you know. So where am I going? Sorry, looks like I need more coffee. Yeah. So hold on. Yeah. So so far I've been while pursuing network level feature extraction techniques to improve the biomarker performance. We utilized a number of machine learning techniques, like I was pointing out before in Sanjay's talk. Instead of just putting them all into a single big classifier, it's actually important to think about which subspace these features lie in, and so we can optimize individual predictive models or kernels. For example, make use of something called multiple kernel learning to not only improve the performance with a composite classifier, but also do so in a interpretable manner to help explain the predictions and go back to where the predictions are coming from. And it's in this process, it's important to actually put the classifier to a rigorous test, like Sanjay was pointing out, to ensure not only test sets actually exposes the classifier to full variability in data, but also do so in a way that we can actually help understand whether the predictions are coming from actual signal related to the disease or the problem at hand, or if it's actually a fluke. So doing good cross-validation is actually important too. So we try to demonstrate the utility of our methods in very different but related applications, especially like early detection of Alzheimer's disease, as well as differential diagnosis in terms of Alzheimer's and frontotemporal disease. And we study a number of other diseases too. So if you're interested, ping me. And replication is a big part in terms of ensuring that the results we see are not just an artifact of a particular data set are reflected demographics in the data set. So we replicated our methods on independent data sets from actually both in different countries as well as in different diseases. So in this process, basically, I realized that the two or three important challenges that are holding the entire field back, not just my method, not just my methods. Essentially, they fall into two big bags or three bins, like I show here, in terms of basically lack of quality in both intermediate and as well as input data, and lack of sufficient validation for methods and software, like I mentioned before, and lack of collaboration, so to say. You know, it's improving a lot, but it's nowhere near the levels we need to actually make real progress on important tough problems like biomarkers and predicting, let's say, doing the early detection of Alzheimer's disease that makes a difference in patients' life, right? So what I mean by that is, when I say lack of quality, what I mean is people don't often look at their data sufficiently enough or closely enough, you know. So quality control either is not done at all often, or when it is done in such a cursory manner that it actually is not effective, or it's probably not helping, you know. We can spend like hours on this issue, but I'm going to quickly skip over and let you ask me questions later on. Essentially, as I try to basically say, how do we help make it easy for people to do this tough task of QC, as well as what's actually preventing them? You know, I don't want to imply that there's people don't care about it. People do care, but it's just that it's a tough, resource intensive, and actually a painful task, to be honest, to do. And even when we set out to do by some labs, there are some lot of open questions in terms of, you know, how do we do it? You know, for example, if we talk about fMRI data, what stage do we do it? Do we pre-process it before looking at it, or do we look at the raw data? You know, what steps to be applied, what parameters to choose, what level of motion is acceptable, and you know which applications it does not matter, right? So there's a lot of open questions as we try to get people to do QC. So in order to help answer, you know, we got together as a special interest group under OHPM, and we actually ran a survey that helped define the scope or framework to pursue this research, which is one of the core directions I'm trying to pursue at the Open Minds Lab here in Pitt. So QC, people often think about it as somehow like an artifact they see in the image, but it's actually, it expands much further than that. Starting with actually controlling the environment or climate, so to say, and hardware in the imaging facility, followed by calibrating the scanner, as well as performing daily random corrections or monitoring, and followed by actually, this is what probably most people are familiar with in terms of ensuring that scanner is in a known state, and actual data QC, like I mentioned before, to make sure that images don't have any artifacts that make them unusable, such as ringing, motion, and other physiological artifacts, right? So moreover, like Sanjay was mentioning, you know, that multi-site data harmonization is actually super important, especially in the age of large open data sets like ABCD or ADNI or Biobank, for example, when there are multiple sites, multiple scanners, as well as multiple types of confounds involved in pooling the data. So I give this to just to give you, not to scare you that it's a huge problem that you may not be able to pursue, but just to give a sense of why it is really complex and why people haven't been able to solve. And we're trying to basically approach this problem in like small doses, so to say, in terms of, for example, catching the artifacts right during the acquisition themselves, so they don't get unnoticed later on when people don't look at it, So one of the things I'm trying to do here at PID is to develop the assistive technology for technologists, so they can actually, it makes it easier for them to spot the artifacts right away during the acquisition themselves, even the complex ones that require advanced and customized visualizations, and also target certain image quality metrics to develop automatic detection of artifacts too. So if you're interested, just let me know, we can discuss more. So this is the first of the big, 2-3 big challenges that I mentioned that is holding the entire field back, in my opinion. The second one is actual performance evaluation. Let's say you did great quality control, you extracted very useful features. It could be any number of things that we actually saw this morning, including the connectivity and cortical thickness or FDG uptake and whatnot. But once you have such a biomarker candidate, how do we know that it's actually useful, right? So like I mentioned, generalizability is actually tough because often you're not learning the full changes caused by the disease because you don't have enough samples, right? So due to the nature of how peer review works in academia and the focus on trying to basically try get a performance number that gets them published, people over the years have tried to basically just focus on trying to get better accuracy numbers, pushing all the knobs or turning all the knobs or pushing all the buttons they can, and that lead to basically not only reproducible results but also basically unusable results, in my personal opinion. So to help solve this issue, basically what I try to do is develop and we can see this what you call turning all knobs approach reflected in the nice review presented by Wu et al. Basically, even in the Alzheimer's domain, where large data sets are available and a lot of people are working on it, you can see large variability in the results, and that is also reflected in other disorders too, going from Parkinson's or depression or many other disorders, even with large sample sizes, right? And another what you call a representation of this problem is in how most clinical trials in the AD world failed because of these what you call so to say cheating approach, and to put it really plainly, right? So I think to solve this problem, it's very important that we have actually a common open standard for performance evaluation that everybody can rely on. So we are measuring the same thing when we compare, right? So this is why I try to develop a tool called NeuroPredict to help solve this problem. Basically, at the core, what it does is take a bunch of features or biomarker candidates, you can call them. It could be anything from different neuroimaging modalities or non-imaging data as well, and produces a comprehensive estimate of its performance based, while implementing the best practices in machine learning, cross-validation, and everything else, as well as software engineering, right? So it's not just, it evaluates not just performance in terms of accuracy, AUC, but also many other important things in terms of biomarkers, in terms of where the biomarker makes mistakes, for example, the misclassification rate, confusion matrices, which features are actually contributing to the prediction, etc. And another related problem, like we learned this morning, is while you make prediction, for example, there is a lot of confounds, right, that you need to take into account. It could be age, gender, site, and many other factors. And accounting for that actually is not possible in my knowledge, even in scikit-learn or other biostatistic tools I mentioned. So it is actually not possible based on either you are adding covariates to the model, like Andre was suggesting this morning in his talk, or residualizing their influence in the features. This is actually one of the important things. So especially in the clinical world, covariates play a big role in the type of questions researchers are trying to ask and answer. So we can also predict not just classes and categories, but also continuous scores that are important for psychiatric applications, So this is just a tool in terms of the larger goal of basically standardizing the performance evaluation. I think what is needed to actually achieve the standardization across the field is this whole framework. I don't think I have time to get into. Essentially, one thing that helps solve this large variability in the field is to have not only an open tool for performance evaluation, but also a comparison portal right at the peer review stage. So one of the instead of just trusting the author, the results in their table 3, that their method is always somehow better than whatever they are else reporting, right? Which is not often the case in my opinion. So having this open comparison portal where it deposits the comparisons into a centralized repo based on openly validated tools would enable us to actually make real progress in terms of biomarkers. For example, in the ADDI dataset, for example, came out in 2008 or so. And even today, after 10, 12 years and thousands of papers, we can't definitively answer the question, when is hippocampal features useful? How accurate are they? When are they not useful? For example, we have some sense, but we don't have a sense that is actually backed by a data-driven quantitative analysis, for example, based on meta-analysis, right? So my hope is to try to build this framework to develop this rich meta-analysis database coming from a centralized repo. Then we can mine it to really answer the question over time. Let's say we have this ready today, and then next few to 5 years, people publish various analyses of their various interesting feature extraction methods, and we can mine this meta-analysis database to definitively answer the question, which is the best biomarker and what are their properties, and in terms of especially varying confounds or covariates, etc. Right. So I had to develop several libraries to make this happen. For example, like the QC problem I mentioned, I think one of the good ways to approach that, because it's a complicated problem, is to develop assistive technology to make visual QC easier, which is what I try to do with visual QC and NeuroPredict I just mentioned. And we'll also talk about this confounds library in the project, which is basically, like I mentioned during the Andre's talk, we have some methods that seem to perform well, but we can't really, in my opinion, they haven't been thoroughly evaluated enough, especially in terms of informatics and software testing perspective. And trying to solve that with a open source confounds library. And like I mentioned, distance metric learning is an important interest of mine in the long term. If you're interested, take a look at kernel methods too. So thanks a lot.
Great job, Pradeep. I think one of the things that I really like about your work is the focus on being able to build infrastructure that can, the one ring that'll bind everything in together in a unified place, which is something that, we're still stuck in this homebrew model for building neuroimaging pipelines that, we can't everybody wants to have their own method and their own implementation. Whereas if we had a centralized place where we could kind of compare methods and features in a kind of systematic way, the whole field in terms of trying to build predictive models will move forward faster. So I really like that about your work.
Thank you. Thank you. Yeah. Any questions?
Yeah, I would like to make a comment on Pradeep's contribution as well. You know, as a big admirer of his work, you know, especially, you know, for bringing us, you know, the attention to these different challenges when doing that predictive modeling. You know, he has also contributed providing us these nice libraries and toolboxes, you know, for these challenges. But I also think that he has also contributed in, at least you were the first person that made the proposition to change the terminology that we use in machine learning. And I think that we should come out, you know, from this brain hack just convincing ourselves that we should use that terminology, you know, of going from the terminology training validation test to a more logical naming term in as training, tuning, and reporting set. So I think we should you know, take that you know, a global goal to achieve, you know, during this brain hack.
Yeah, thank you. Thank you, Javi. It looks like I need more coffee. I totally blanked out for the first